Good afternoon, everyone. Welcome to part 4 of our urban rodent control series. Before we get started, I want to go over a few webinar procedures. Please, everyone, mute your line. I've taken care of that on the back end, but if you become unmuted, please mute yourself again. You can use the chat box to ask questions, and all the questions will be asked at the end of the webinar or answered at the end of the webinar. For additional questions not answered during the webinar, you can email us at education@nola.gov. The webinar will be posted on our YouTube channel, which can be found by searching New Orleans Mosquito, Termite, and Rodent Control in YouTube. Today's topic is the importance of managing rodents. It's a comprehensive look at the risks commensal rodents pose to public health and ways to increase preparedness. Today's speakers will be Dr. Claudia Rigo and Bruno Gersi. Dr. Rigo is the director of Mosquito, Termite, and Rodent Control Board. She earned her degrees from Purdue University, University of Georgia, and obtained a PhD from the University of Florida. In 2004, she became Principal Research Entomologist for the City of New Orleans Mosquito and Termite Control Board. In 2006, she became Assistant Director, and in 2010, she was promoted to Director of the department.
Thanks so much, Astacia. And it is my pleasure today to introduce Dr. Bruno Gersi. And he received his DMV from the Universidad Nacional Mayor de San Marcos in Peru as a veterinary research scientist at the U.S. Naval Medical Research Unit 6 in Lima, Peru. He led the avian influenza surveillance program there. He participated in lots of studies on rodent control and bat zoonosis diseases surveillance in urban and rural settings, as well as swine flu surveillance and primate zoonotic disease epidemiology. I actually had the pleasure of meeting him when he came to the U.S. And worked at Tulane University to obtain a PhD in rodent ecology. He's working with Dr. Mike Blum. And then later he transferred to the University of Tennessee when his advisor moved there. And he defended his PhD in 2020. He worked at UC Davis doing a postdoctoral program at looking at Lassa virus ecology in West Africa. And now he's gone all the way to the other side of the country to Tufts University, looking at how to stop the spillover of zoonotic diseases to humans. So we're really happy to have him today. And so I'm going to start off a little bit, and then we're going to transfer. I'm going to kick it over to Bruno, and then he'll kick it back over to me. So look, I think the main thing is for everybody, this is City of New Orleans Mosquito and Termite Control Board. We've been around for a very long time. We've done mosquito control, but rodent control used to be at the health department many years ago. So that's what we do. And I think, I like to read history. And so in lots of historical books. And so I think we need to go all the way back to the time of the Romans. And I think, when we think of it with the Roman times, perhaps in movies and all of that, we think about, the folks that were in charge. But the everyday person lived in a very crowded environment with lots of ectoparasites, fleas and lice and mites. Rodents were common every day. Flies and mosquitoes and bed bugs. So humans have been living with all kinds of different diseases for many, many years. And I think, one of the most famous examples, of course, is going to be the Black Plague. And so again, we need to read history, learn from the past, and hopefully not repeat some of the things that we've done. Estimated worldwide, over 200 million people died, the 1300s pandemic. About 25 to 33% of the populations died, right? So really tremendous. So look, and many of us maybe don't think that there is current plague around, but there is. And this is just a map of the world looking at different sort of cases of plague that is occurring. This is the source here is the WHO, but there are animal reservoirs for plague. Some mice, camels, chipmunks, prairie dogs, for example. Colorado has problems with that. Rabbits and squirrels, of course, and fleas as well. So very important is the oriental rat flea. Even in New Orleans, there's unfortunately a very rich history of lots of diseases in New Orleans. And one that is really resurfaced across some parts of the country is murine typhus. And, it's really interesting because New Orleans had Charity Hospital, and they recorded cases of people that came in with different diseases. It may have been yellow fever, murine typhus. So there's a good record of all of these. But many years ago, in the 1940s and 50s, they were doing surveillance. You can see that there were cases of murine typhus in New Orleans. They were also looking at the rodents, tracking people. And in the 1970s, there had been an absence of murine typhus, but increased rodent populations, lots of different workers, and they were looking at the rodents as well. And you had 40%, almost 41% of the rodents tested positive for murine typhus. So there was a big resurgence, and there was a big program in New Orleans in the 1970s looking at, actually it was treatment of the rodents, so basically control, and also doing work using malathion to kill the ectoparasite, right, which is the oriental rat flea. So it's just really important. I am always fascinated with this. 100 rats per hour, that's a really high number. We don't see that anymore, not even close, but that's incredible. They used approximately 5.5 tons of bait, and they also used, the malathion for the ectoparasites. But with all of that work, finally in 1972, they had 0 cases of murine typhus, and actually that really helped expand the program of the rodent control program at the city of New Orleans. And so when we think about the urban rodent problem, right, it's contamination of food, it is destruction of property, it's disease transmission, it's increased asthma cases, and it just doesn't look good for a city. No city wants to be known as the rat capital of the country. And so there's lots of reasons for this, right? And if you cannot think about it from a worldwide standpoint, or under even in your area, your jurisdiction, or the area that you work in, we are constantly encroaching on those forests, right? So that's an increase of people and in those forested areas where these animals are. We have population growth in many cases, urbanization, lack of infrastructure, and in some cases very poor infrastructure or old infrastructure, increased levels of poverty, poor levels of hygiene. Access to healthcare might be a problem in many places. I mean, we live in a city, but even within a city, do you have access to a physician? Resistance to pesticides, it can be an issue. There may not even be enough expertise or training in vector control. And I'm going to talk a lot about preparedness after Bruno does his section. And really the state of rodent control across the country is not great. We need to get our public to participate, right? So that might be restaurants, that might be, to clean up, or just in the news recently, there was a chain of stores that had a terrible infestation of rodents, and there was a large recall. We need to do better to work with our architects and builders to build out rodents from the get-go, right? And of course, climate change and increasing temperature might expand the range of some of these pathogens and include, and also some of the rodents that we have. Why do they make good agents for disease transmission? Well, there's a lot of diversity in species worldwide. Many are incredibly opportunistic. They have high reproductive potential. The high population densities can also occur in many species. And very important here are these peridomestic affinities. So they're in and around our properties and where we are. And so, we have to think about how transmission occurs between animals and people. And these are 4 major ways. And guess what, guys? Rodents fit every bill. So let's talk about direct contact, right? So it's really coming into contact with saliva, blood, urine, mucus, feces, or other fluids of that infected animal. So as all of you are doing your inspections, it may be a restaurant inspection or a building or a house, and you see rodent droppings, right? So that's coming into contact. So indirect contact as well, going into areas where they live and they roam, so you're touching those contaminated surfaces. So imagine being in an attic, for example, in a closed space. So that may be an issue. Also vectorborne, right? So there are fleas. We just talked about murine typhus. So fleas may also be carriers of particular pathogens, and foodborne as well, right? So you can have rodents that are on food, contaminate food, and there you go. So it's a potential for becoming sick. I also think it's important for us not to forget about allergies, right? Which is an overreaction of the immune system to substances that generally do not affect other individuals. Mice are very important when it comes to allergies, particularly in kids. Symptoms can become sneezing, coughing, itching. Some folks actually have chronic conditions, unfortunately. And allergies, allergic reactions can range from very mild to life-threatening in some cases. So we've got rodent allergens are very, very important. This is just a publication, some graphs from a publication, but it just show you asthma levels can increase with mouse allergen levels, right? And this is it with kids in schools. And unfortunately, as those allergens levels increase, the lung function of these kids decrease as well. So it's very important that we pay attention in schools. So I'm going to kick it over to Bruno. He's going to bring up his presentation. Let me go ahead and release. There you go, Bruno. I'm going to should be able to pull it up, and he's going to talk about some of the work that he and Dr. Anna Peterson have done in New Orleans.
As usually with these things, I start talking when I'm still on mute.
Yeah, you're not on mute.
No, I'm not. Thank you, Claudia, and thank you guys for participating and spending your time here listening to me ramble a little bit about the work that Anna and I did as part of our dissertations for our PhD while we were both at Tulane and Tennessee later on. We definitely couldn't have done any of this work without Claudia and her team that help us in so many ways on getting access to the community and also on the field. So again, I'm going to be talking a little bit about rodents in New Orleans and their pathogens. And does this one do? Does this go? Okay. Handle. So as Claudia mentioned already, why do we care? Why do we study rats, or what's important? They are, as mentioned before, widely distributed. They are on every continent except very cold environments like Antarctica. But as climate changes, it might change, and they might get there. They are very cosmopolitan. They are going to be in very close relationship with humans. So every big city has rats. After that time I've worked with rats, and I'm sure it's similar for all of you, once you start recognizing tracks and marks and burrows and so on, I don't think there's been any city I don't go that I see either I actually see a rat or I see marks that rats and rodents are around. There's a very strong cost to damage of property, and they make news apparently a lot. Sometimes just as bloopers about like rat pizza, or sometimes in cases of more important things like damage and diseases that they transmit. Here are some photos that I've took. Some are actually from my property. The chocolate bar was in my house and the mice came in. I lost that. And you can see damage that they do to car engines when they're through the cables over winter, when you leave your car standing for over a week, unfortunately. You can also talk about the diseases that they can carry, and we're going to be talking more about this in later on, but rodents are reservoirs to 46% of the major zoonotic diseases that humans have. We can talk about hantavirus, leptospira, plague, as Claudia mentioned. And to prevent these diseases, we need to understand transmission, and to understand transmission, we need to understand the host. How do they get to us? How do they get close to us? So that's important. That's why we need to understand rats. So talking a little bit of what the research is and how we came to do this. My advisor got a big grant in collaboration with Claudia Regal and others, looking at the effect of Katrina and post-Katrina management on rat populations. So we, as part of this study, we went to 10 different neighborhoods or areas of New Orleans that had different land management strategies that were areas of high income, low income, areas that got flooded during Katrina, areas that didn't got flooded, and so on. And in each of these areas, we went to 10 different city blocks. Why did we choose city blocks? Mainly because rats, although they've been proven to walk up to a kilometer a night, they don't get far from their nest, more than 100, maybe 150 meters from their nest in a regular night. So a city block was a good size of identifying straight rat communities and what was in that area. We went to these sites, we set up tomahawk traps at either personal back the backgrounds of houses or their yards, as you can see in the photos here, or sometimes it was abandoned lots that would be managed by the city or just not managed if they were still owned by a private person. So on, we would set up these traps and check them, leave them for 3 consecutive nights, checking them twice every morning, setting up at night at regular as usual. And we would go back to these same blocks and hopefully to these same lots twice a year. We did this for 3 years. And as you can see here, you can compare city blocks in Uptown New Orleans here on your left versus the lower Ninth Ward, that it's the area down here. I'm not sure if you can see my cursor on the photo on the right, and the aerial images of the same city blocks. Once we got to a block and once we got access from the owners, if there was anybody to get access, we would take information about overgrown vegetation, presence of debris, trash, presence of burrows, if we could see them, or uh, runs from the rodents, mainly to be able to complement this information, how they compare to actual presence of rats. As I mentioned, traps were set at dusk. We would be always wearing gloves. We don't want to scent our traps with our smell. We would use peanut butter and bacon as our bait. And early in the morning, we will collect any traps with rodents and did a full processing of the rodents to collect tissues and biometric information to test for different pathogens, so on. Over the 3 years of sampling, we had a trap effort of 50,500 trap nights. That it's a very intense work when you consider that it was a team of 6 or 8 people doing this. And so, okay, we collected rats, and what did we find out? We found that rats are, as expected, all over the city. We capture rats in every neighborhood where we set up traps, not necessarily in every block, but at least in every neighborhood. And we were able to notice that overgrown vegetation, that it's a problem when there's abandoned lots and abandoned areas of the city, it's a very good predictor of how many rats you will find in that area. We overall found 5 species of rodents, not only Norway rats and roof rats. So we found both, and New Orleans has both species of rats. We also have mice, or the common mice, and hispid rat, and another native rodent from the area. And these were all found in areas that were less urbanized or abandoned of the city. And you can see that the abundance of rodent and the richness was related to the counter urbanization. So areas that were less urbanized had more rodents and more species of rodents in them. And again, rodent abundance was greater in residential areas, burned with vacancy, and with other conditions associated with vacancy as debris, the abundance of debris piles, as shown on the photo, and the overgrown vegetation drove population size of rodents in the area. As I mentioned earlier, we had both rats in New Orleans. When we started this project, we expected to trap only Norway rats. So all our traps were placed at ground level. But we ended up finding out that actually New Orleans has more roof rats than Norway rats. So we found Norway rats on only 38 of the 96 sites we visited, while we found roof rats in 71 of those 96 sites. Roof rats were more widely distributed around the city, and they were found in higher numbers than Norway rats. The only neighborhood that had higher numbers of Norway rats versus roof rats was the French Quarter. And if you've been to New Orleans, you know that this is a very commercial area, very touristy area. So there's a lot of restaurants, bars, and with them comes trash between what the people drop and the amount that the restaurants produce, and open containers of trash and so on. We looked at different factors and age distribution of rats over time. And for example, we noticed that the age distribution of Norway rats was even over time. So the same number of percentage of juveniles versus subadults and adults over the 3 years over summer and winter. So there was no difference. We relate this to that, as I mentioned earlier, they are in areas that are more commercial, and they depend more on the food that humans provide, mainly trash. So they don't see a difference in food sources over time. In comparison, roof rats, you would see an increase on juveniles over winter. That means there were more subadults during the summer. So it's more related, it might be more related to vegetation. And in New Orleans, many people will have vegetable gardens or fruit trees in their property. So you would see the roof rats coming to the fig tree or the lime or orange trees and picking on the fruit. And this is also a risk for transmission, as Claudia was mentioning early. We also, as we were collecting all these samples, we looked at the genetic structure of these rodents and looking at their populations. And Why do we care? Is cities in natural environments, you would have natural barriers as streams, rivers, that will limit the movement of individuals and communication between populations, as you could see on the upper left graph. But sometimes there will be animals that are able to cross that barrier, either because they are better swimmers in this case, or they are faster, or there's a tunnel underneath. If we look into convert what a river would be to a highway or a two-lane street with high traffic, so on. And you can look at how much of those genes are being shared between populations and how much individuals move. Why do we care? Mainly because this helps us understand how big of an effort we need to do in time of how many blocks we need to treat if we want to eradicate populations, and how long would it take for those populations to come back if they start migrating back from other areas that weren't treated or controlled. So New Orleans, in the case of New Orleans, we could identify 5 different rat populations, genetically structured. If you see, compare the map against the PCA graph, you can see that rodents that came from the French Quarter and the CBD area are genetically distinct from rats that came from the lower ninth ward, the green circle here, that are different from rats that came from the Bywater and upper ninth ward area of the city. And you see that there's some level of gene crossing or sharing between lower ninth ward and upper ninth ward kind of locations. In between the lower and upper ninth ward, you can see that there's a water canal, so there's a very strict barrier, but there's still flow of trucks and vehicles, and there's bridges, so there's still some movement happening there. Again, looking at the genetics, you can see how the structure comes, and this is a different graph showing the same results, that we see 5 genetically distinct populations. Now, when we look at how related each individual each to the next in relationship to distance between from where we collected one to the other, this would be like comparing families that never leave their town against families where the cousins and the nephews start leaving town and going to different cities, and then they're spreading genes and sharing. You would move the same idea into city blocks. And when you analyze the whole data set as a whole, in New Orleans, you would see that rats share some genetic similarity up to 2 kilometers apart. So you can identify third-degree cousins up to 2 kilometers from one colony to the other. When you separate this information and look at the different areas of the city, as I mentioned, there are different colonies and genetically they were different. So looking at the rodents that came from the lower ninth ward and the CBD area, you can see that difference or that disconnection between how closely related they are goes only for up to 250 meters. So one city block, and then you have a completely different colony, different genetic structure between them. Sorry, in the French Quarter. In the lower ninth ward is a little bit more, and so on. And again, this helps you understand and develop the strategies for pest management. Now, as Claudia was mentioning earlier, we have plenty of diseases. As I mentioned, there are 46% of major zoonotic diseases are come from rodents to humans, not necessarily Norway rat or roof rat, but from rodents. And we wanted to see which of those zoonotic diseases were present in the New Orleans rodent population and what the risk was for transmission to humans. So I'm going to be talking of a couple of those diseases. I'm going to start with Chagas, that it's a parasitic infection. It's transmitted by the kissing bug that you can see on this image, and probably you've seen this insect when doing pest management. Globally, it's an estimate of 30,000 new cases a year. It's can be symptomatic, and symptoms will go from swelling of the site of the bite of the mosquito, the vector bite, or it could be asymptomatic for a while, and then you start developing heart issues, esophagus, colon, digestive problems as your muscles start losing strength. There are 11 species of the kissing bug, and that can transmit the Triatoma cruzi, that it's the name of the parasite. And 11 of those and it is present in the United States. It has been reported in wildlife, but not much in urban areas. There are other studies that have shown presence of Chagas in bigger mammals, in raccoons or other mammals that also are around cities, but they move far. You'll find them in your blog, but they will go kilometers a night. The big difference with working with rats is that, as I mentioned, they don't move far. So if you find the parasite there, that means that the vector is there and the potential of transmission is actually in that block. So again, we used the samples that we had already collected from the ecology part of the study, and working with a professor in the public health department from Tulane. We took samples from the first 2 years of work and test the blood for presence of the parasite. And we found that 11% of the rodents were positive for T. cruzi. And again, if you can see the map, you will notice that it's around all over the city. So you can see the number of positive individuals and how many of those were infected with the parasite. And you can see again that it was present in Norway rats, roof rats, rice rats, and other rodents in all the different neighborhoods that we sampled. Except the CBD, that it was very low. But again, the central business district doesn't have much vegetation. We then noticed that the prevalence was higher in areas where we had more than one species of rodent. And we also noticed that the amount of vacancy or empty abandoned lots in a city block was a good predictor of the infection, the probability of the rodent being infected. So where there were more abandoned lots, the prevalence would be higher. And this was dependent on the season of sampling, like winter versus summer, as you can see on the graph. We also tested the kidneys for presence of Leptospira, that it's a gram-negative bacteria. Leptospira has multiple strains, more than 17 different species, more than 200 serovars. Some of them are pathogenic, some are not. But again, transmission of leptospira is from the rat. They will excrete it on the urine, and it will go to the soil. And then, if on water, if you walk and have cuts on your feet or something, the bacteria will get inside and infect you through that route. And again, we saw a prevalence of 15% of the rats that we collected. Again, we saw that the bacteria was present in all neighborhoods where we sampled and tested. And it was distributed, it was present in all the species of rodents that were in the area. One interesting factor was that different rodents had different strains of Leptospira. So Leptospira interrogans is one of the most known and pathogenic, and it was very common on the Norway rat, while Leptospira borgetensis was present on the mass. Either way, both are bad. You don't want to get exposed to it. So it's another more reason to be careful when handling rodents or being around areas where rodents are around. Going back to parasites, now a nematode, Angiostrongylus cantonensis, or rat lungworm, as it's also known. You can see the worm, the photo of the parasite on the lower left. And these are present on the lungs of the rat. So the adults will be on the lung. The eggs are expelled on the lung and swollen and go on the feces. And the intermediate host is the snail. So people get infected when they eat unwashed vegetables, or when, as some recent case happened in Australia, somebody to eat a raw snail just for fun. So again, these parasites have been detected in United States. It's been reported in Florida, and we look at the presence in New Orleans, and 38% of rats sampled had the parasite, so it's a very high prevalence. There was some variation on prevalence between neighborhoods. As you can see, the lower Ninth Ward had a very high infection versus Lakeshore and Lakeview that had a lower one. There was some variations between species, so between Norway rats and roof rats. And obviously, as it's an infection that it's chronic in the rat, the older rats were more likely to be infected than juveniles. Another bacteria is Bartonella. There are different species of Bartonella. It's a gram-negative bacteria. You might have heard of cat scratch disease or trench fever or carrion disease. These are three of many diseases that are caused by different strains or species of Bartonella. Bartonellas are transmitted by fleas, by vectors. Some will be transmitted, for example, cat scratch is transmitted by fleas. Trench fever is transmitted by lice, and carrion fever is transmitted by sandflies. So we looked again at Bartonella in the rats in New Orleans, and we found that 13% of rats were infected. We only tested samples from 1 year, so not all the samples we collected, but 40 Norway rats out of 160 were positive, and these 40 rats came from 2 sites, while only 5 roof rats were positive for this bacteria, and they came from 5 different sites in the city. As I was mentioning, fleas are the main vector for this, so flea infestation was a strong predictor of if the rat uh, will be infected or not with the parasite. And I'll ask Claudia this later, or we can confirm this, but if I'm correct of this, most of these rats came from a homeless encampment in the city. So that shows you how close humans and rats are, and how vulnerable homeless populations are to these diseases. So what have we learned today, or I hope you've learned, is that no city is safe. There's rats in every city and they are all over. Debris piles, garbage, overgrown vegetation, abandoned lots, and abandoned structures are factors that facilitate the abundance of rats. Rats will disperse or be related to up to 2 kilometers, but it will change depending on their environment and their needs. In areas where there's enough food next to where their nest is, they will not move far, but it might change. Chagas, Leptospira, Ratworm, Bartonella, these are some of many zoonotic diseases that rodents can have. And if we look for them, it's most likely we will find them. They are very common. And we need to consider this when we are doing pest management. If in many cases, when we're talking about plague, for example, and it might happen the same for Bartonella and others, if you eradicate the host, meaning the rat, you leave the fleas around and they will look for the next thing to feed on. And many times that means us as humans. So something that it's good to consider always is if you're treating for rats, might be good to also treat for the vectors in the area. And again, some of these pathogens will be in the soil, will be in the feces or in the urine. So when you're crawling under those houses or on those attics, be careful because you are getting exposed. My main thing is always try to wear gloves and have dedicated clothing that you can then wash and disinfect and not bring home. You don't want to bring any of these diseases to your family and expose them to any of these. As I said earlier, this was a teamwork. I didn't do it alone. It was not just me and Patricia. Claudia and the group at the Mosquito Termite Control Board were very important in helping us collect all these rodents and getting access to all these houses and properties. We had a bajillion undergrad students helping us on the fields, on the lab, and so on. And we wouldn't have been able to do any of this without them and without funding to do all this work. So have to praise them. Also, I have to thank my advisor and the postdoctorals that help us with the analysis and the work and all this. So, and with that, I'll leave it to you guys if there's any questions before I have to hop off.
Yep. Bruno, I think what we're going to do, just for the sake of time, if folks have questions, right, if they can make sure to put it in the chat, and we'll try to address them at the end. I know you need to go to another meeting, but if I can't answer it, we'll make sure that, you know, we forward the questions to you, and you'll be able to answer them directly. So I appreciate you spending the time with us. You know, I think, again, as we sort of go into the next portion of this presentation, which is going to be preparedness, this project really allowed, right, to have some critical information, which is where are the rat species? What is the pathogen loads, right? All of this information that we need to know so that we can, as a city, become more prepared, and how can it improve our operations, right? So that cooperation between university and municipal and state programs are super important. So let me turn it over to get my presentation back on. I think folks can see this now. And I'm going to continue to talk about, I think, again, you know, the whole premise of this presentation is why control rats? Who cares, right? Well, we really should care. And I think it should be loud and clear from Bruno's presentation that if you are working with rodents, you should be wearing the proper PPE every single time, all right? Because if your area, let's say you have researchers or your programs haven't looked for those pathogens, the very likelihood that the situation is that you likely have pathogens in your rodents. All right, so let's talk a little bit about rodent control preparedness, right? So I work for a city. I look at it from disasters, and it can really, I really actually should really consider all hazards. You know, we just recently had COVID shut down, so that created a whole nother scenario, but also we're going to talk a little bit about hurricanes and some other things in history. All right, so this is when, in the context of my presentation, this is what I am talking about, is really dealing with the commensal rodents. So the Norway rat, the roof rat, and the house mouse. And you'll see that there are publications listed on many of my slides. You can take a screenshot and go look them up. But there was a recent publication from Michael Lee. He's from the Vancouver group. He was getting his PhD, and he went around and interviewed all kinds of programs that dealt with rodent control. And then also NACCHO, it's another organization. Some years ago, they went through and they interviewed a bunch of programs as well to look at the situation with rodent controls. And really, it varies very much in size and in scope. And a lot of cities don't have a municipal program, right? And so we really are reliant on our pest control partners to fill in the gaps there. We need to make sure that we have the ability to take information, right? So 311 is someplace or a system that some programs have. Funding levels are all over the place. When it comes to rodent control, public or private contractors, you know, are those in place? The specifications of programs vary a lot. Some just will service a complaint that comes in from the public, and that's it. That's all they're looking at. And others have very advanced and sophisticated programs. Are there outreach programs, right? What are the tools that are out there for surveillance and control? So everything really can vary. Now, the one thing, you know, in rodent control is that a lot of programs work in silos. And so that means, let's say one person's not communicating to the other group. And so very important for people not to work in silos and have good communication. So when I think about rodent control preparedness, these are some of the basic infrastructure and information that I think everyone should have, right? You need to know the rodent species in your area. Do you know it? I hope you do. But I'm not sure anyone has really cataloged and trapped and really understood. So very important. Well-informed and funded municipal or state programs, right? All of us on this call really need to be advocates of vector control programs. It just doesn't happen by itself. You need to really be a good advocate. There needs to be a way for the public to report sightings or infestations. Very important. Is there routine surveillance or periodic surveys? Really need to have that because things change. Even the work that was done by Bruno, Dr. Blum, and team here, that's been several years already. So we need to make sure that, you know, we're moving forward and we're understanding that rodent population, how it changes. Interagency cooperation and information sharing, absolutely critical. Understanding the infrastructure, sewers, right? Underground, what's going on. Buildings and parks, private industry capacity, you need to understand that and to see if it's there. And also what tools are available for monitoring, trapping, what rodenticides are available. In some places they're not. And so non-active baits, pest proofing capacity, all of those control parts that are really integrated pest management, you need to know what is there. Public awareness and understanding, right? So we always are working towards better education of our public. We actually right now have a survey that is released out to our public, so we have a better understanding of our residents and what they know about rodent control. And ultimately, you need participation of that public or that customer. So when you're saying, please clean something up, or stop feeding the birds outside or stop putting pet food outside, we need folks to help. All right. So when it comes to a hurricane, we've had quite a few here, unfortunately, here in Louisiana. But passive immediate concern, mosquitoes, flies, rodents, wildlife, stray animals, right? So these are things that we worry about. Rodents generally are not a problem immediately after a storm, all right? So the number of rats and mice are often reduced after these natural disasters. It could be floods that come in, it could be earthquakes that really damage, but immediately after, they're typically uncommon. They begin to establish again within that 6 to 10 month time period. And there's plenty of data to support that that is the case. That's actually what happened here in New Orleans after Hurricane Katrina. So the role of rodent control, right? Vector control during an emergency, very important for that long-term recovery. There is a role in the Emergency Operations Center for rodent and mosquito control, very important part. You'll find that after these disasters, there is a rodent hysteria, okay, and a lot of sensationalism. So people get very concerned. Maybe they haven't seen a rodent in their neighborhood before or in an atypical place. So it's very important that we're sending those good messages. A lot of wrong information I have found that is often disseminated. So we need to make sure our messaging is clear. And we have to understand the current rodent situation prior to that storm, right? Or that emergency is very helpful in where you're going to be spending your time after. Implement control in those critical areas, partner with agencies and the pest control industry to get the job done, and work very closely with communications to make sure that you're putting out that information that's needed. I know that there are a lot of folks that are in the public sector, so that's why I'm putting this information here. But you can read the Stafford Act, and we are vector control, mosquito and rodent are considered Category B eligible. Now, there's a bunch of rules and regs on that. It's not for today, but you should, when you have some time, look at the Public Assistance and Policy Guide. The other thing that's important that everyone should check is, and this was very important after the COVID lockdown, is that pest control in Louisiana was considered essential service. So we never stopped, right? There were states where pest control was not considered an essential service. And so many health departments, folks that were working in this area weren't able to get out in the field and do the work. So I think it's really important. I know a lot of work has been done since with the National Pest Management Association on this, but I think everyone should check their state and make sure. And if it's not considered, if you're not considered essential service, then I think you really need a lobby towards getting it that way so we can do the work. So you already heard my first part of the presentation. I like to look at history. There's lots of great information. And so let's just a couple of slides here looking at in the 1900s to 1904. Looking at plague. It was the first recognized plague epidemic in the US. And really, it was to be honest, poorly handled, in the very beginning. And so San Francisco actually had an earthquake in 1906. And guess what happened here? It took a few years to get plague under control. But then in 1906, here comes a terrible earthquake, a 7.9 magnitude, and the population of rats boomed after the earthquake. And so again, they began to see more plague cases, but this was much better handled in this situation. And actually, Dr. Rupert Blue here ran the anti-rat effort. And, it was really interesting because what happened? They worked on sanitation, they worked on trapping, quarantines, like all of the things that should be done in order to stop an epidemic. So super interesting. I was at the bookstore just this weekend, and there was a new book. It's from 2019. I haven't read it yet, but I thought I would show it here because I know there's a lot of folks from California, but many of those photographs that I've showed were in this book. So I think it's going to be a really interesting read and quite in depth. So when you look at hurricanes, right? So we've learned a lot since Hurricane Katrina. This is 2005. This is actually my photograph flying over the city. And you have to be prepared to come back to this. You know, how do you do your work when your facilities are devastated, damaged, you've lost many of your things? And so again, back to preparedness is using that integrated pest management approach from the very beginning, even prior to the disaster, is having everything already in place. It will make a response more effective, it will run smoothly, and you will really likely do a more effective job. So rodent inspection, we're going to cover a little bit of each one of these components. And so this is just a slice in time, but we want everybody to think about how can your public in your area, and then we do have pest management professionals on the call, how can they reach you to let you know that they have an issue, right? So we happen to have a situation where people could call us, call our building prior to 2013, and then after that, the city implemented a 311 system. We helped them work on the script and all of that. So when there are problems, we're able to work with our 311 system and already give them scripts of things that we think will happen, or questions, frequently asked questions that we can push out to the public. And again, working with Bruno here, very important is for you to understand the rat species across your area. So this is very, very helpful in just general management. And it's exactly the same with knowing the different types of pathogens you have and where are your high risk areas. Okay, and so this just happens to be a map with Bartonella. There's a publication at the bottom if you would like more information. Now, I cannot stress how important sanitation is, right? Sanitation is rodent control. And after a storm, this happens to be after Hurricane Katrina, there may be, because it was so severe, there was a breakdown for many, many, many months in trash collection, right? So sanitation, trash, eventually you're going to get rats, right? So these are the things that you should expect when you have a devastating storm. And then the other thing is the trash changes over time, right? As people are getting in, they're taking out the refrigerators, their wet carpet, things get piled on the sidewalk. And there is really a challenge. This actually happens to be after Hurricane Ida. And so again, we need to work with our customers, if you're inspecting restaurants, that this is just not okay. And so there needs to be good trash management on a daily basis. The more prepared you will be, it really depends on how prepared you are today, prior to that storm. So we created some business and apartment guidelines for people that I think is pretty interesting. And we looked at, again, this is in cooperation with our state sanitarians as well as our Department of Sanitation, and just reminding people of what are the rules, right? Sometimes they just don't know. And then, of course, creating educational information that proper dumpster management is rodent prevention. So really important for us to get this message across, because it will play a role later in preparedness. Exclusion, right? So simple. Here is a run from Mississippi River to an open garbage can. So this is a little bit of an old photo. These areas have been renovated and these trash cans have been changed, which has really helped this problem. So again, the better you are at dealing with the issues today, the better you will be later. And so garbage cans were introduced in 2007, the closed cans. We need to continue to work with our customers to say here, and that's our residents, to say, look, you got to keep that lid closed, right? So an open can like this is food for rodents, but this also made a big change. Remember, Katrina happened in 2005. And at that time, the garbage cans were open. And I remember, I've been here for a long time, I remember going from house to house to house with rat burrows next to open garbage cans that we had to deal with. So just this change has really helped us tremendously. In 2004, our RTAs, or our bus folks, transportation, they started putting these closed can systems at the bus stops. Really important. 2015, our sanitation department began to convert open cans to closed cans. Now, we still have a long way to go. Okay, these are very expensive, but this is a really good thing because it is eliminating the food source from these animals. Trapping and rodenticides are part of control, right? But you have to follow the label, use these products properly and at the right time. So this happens to be Timmy here, Midair, one of our rodentologists, and this actually, and Sean is right here. This is actually after COVID-19 shutdown. So people were not on the streets in the French Quarter. Rodents were actually coming from underneath the doors. We actually had tried some storm drain baiting, but that didn't work so well. So in this particular case, so we ended up going with bait stations and doing treatments on these very busy thoroughfares that were very quiet at the time. We have been incredibly successful with this. We've been able to push the populations of rats down. This is primarily our Norway rats area. And we are still reaping the benefits from being able to do this work at that time. After Hurricane Katrina, remember I talked about the 6 to 10 months, open garbage cans, right? So we started to see elevated numbers of rodents at that time. And so with some volunteer groups, we were able to actually bait the storm drains of all these areas that were in yellow. And we were at those sites for at least 2 months. And it wasn't you just put the bait out and don't ever come back. We actually went back and we looked at bait consumption in these areas. And the main thing with rodenticides is that if your rodents are feeding on it, you need to make sure that you give them enough bait. That means you have to service those stations at adequate frequencies so that you're able to eliminate those populations. So just to give you an example, please keep good records. You will need to go back. You will want to look at your data. This is a long time ago from 2005. A lot of folks have just gone electronic, but regardless the way you can collect your data, you need to make sure you take good data. Trapping is very important. We don't have time. You guys have already watched some of our previous webinars. We'll be posting those on our YouTube channel that talk a lot about trapping, how to trap, what to use, and just a lot of tips. But this is a very important part of control. So that's where you can find that information, as well as education and preparedness. Know who you need to communicate to. This is my group of people that I communicate to. Residents and decision makers and all of our partners and stakeholders, all these folks are included. And you will need to have strategies for each one of these different groups. Do not do this in an emergency. Take the time and prepare yourself now with your messaging and where are these different groups of folks, your target audiences that you need to reach, and how are you going to do that? So clearly after an emergency, you're not going to be in a school. And now that, hopefully we're getting back into schools, this will be, again, part of our educational strategies. We work a lot with the pest control industry. There are a lot of folks that have no cell phones, or it's very basic. They don't want to get their information on social media. And there's a whole group of people that's the only way they want to get their information is on social media. And again, there will always be a group of folks that they want zero pesticides, no matter what the situation is. So you do need to learn how to work with those groups as well. And remember the silos that I talked about, right? So in rodent control, we need to make sure that that doesn't happen. And so you have to take a multi-agency approach. In my case, it's Centers for Disease Control, the Department of Health, different agencies in our city. Prepare your press releases in advance. It is hard to do these things when you're in an emergency. Trust me, okay? I know I'm talking from experience. And then I also want you to think about what are the different things that you need to have in your shop, in your business, so that you can quickly react. Your education for the public is a very important component. You need to make sure that you're producing a unified message. And you have to keep it simple so that everybody understands. This is just a great example. This is from August, maybe September of last year when Ida rolled through New Orleans. there was just some issues with trash collection. And in many places, trash hadn't been collected for almost 2 months. And so we worked through communication on a simple message on spraying bleach inside of your bag before you put it outside. That's the one thing that will truly repel rodents. They do not like it. So these are just some ideas that we have here on education. So you need to work on your education now. And it may be that if you're a pest management professional, you're working on your customers, right? You're giving them information. If you work for a state, your communications is going to be at a larger level. If you're working for a city in a department like ours, you're really talking to your residents. And so these are things that you can do now in order to help. And then also understand how you can reach everyone. So social media is very powerful. There are often communication groups in cities, and we're able to reach with a couple of strikes on a computer over 100,000 people. So pretty important. I cannot stress enough on this preparedness is that you know who your partners are. If it's the health inspectors or city sanitation department or whoever your partners may be, you must know them now so that when you are in an emergency, this happens to be after COVID, we wanted to make sure that these properties didn't leave all this trash around. So we called our partners and issue resolved. And again, your partners may also be landscapers and other folks. And so in this case, you've got a palm tree that is overgrown with lots of the nuts on there. You can see a pigeon at the bottom, but this is just food for rodents. And so this needs to be managed before. Again, don't forget about pest proofing and doing the right thing here to remove your rodent conditions. This happens to be the French Market in New Orleans. Just an absolutely fantastic example of cleaning, making some structural changes so that you do not have issues with rodents. And again, on a preparedness level, you've heard a lot from Bruno already on diseases, and this is part of it, is understanding and what are the problems and what are the pathogens. And so again, if you're a pest management professional, I know most people are not doing this work, but you really need to start thinking about, are there universities or am I able to bring this capacity in-house? And there it is. We actually are trying to create a true surveillance program. We've got a molecular biology lab and professionals that can do this work. And so now we're just setting up, step by step, and so that we can do surveillance of pathogens in-house. Bruno did mention the homeless encampments, and I think for many areas around the country, this is a big problem. You cannot neglect it, okay? And I'm going to give you a couple ideas here on what you can do. We actually avoid using rodenticides entirely in these areas, and we've made significant strides. Use pest proofing. We also use rat ice, which is carbon dioxide in dry ice format, but there are also tanks with dry ice that Leaf Attack sells in part of the country. Look, a simple use of concrete closing up holes works. And so it is not a nice job to do, but you may have to do it. And this is a great way. This is an elevated platform of the street. And basically the rats were entombed inside. And so it's not a once and done. You have to put this out, close the holes, but come back and see what's there. So the use of carbon dioxide is a great tool. It's fast, no residuals. It's not that traditional use of rodenticides. It can use in a variety of places, but really important is it kills insects. So I think all of you heard Bruno say that if you kill that rat and there are people close to it, you're going to need to worry about those ectoparasites. So this is how it works, guys. This happens to be rat ice. They're pellets. We're wearing gloves, right, because it's very cold. Carbon dioxide is heavier than air. So you put some on the burrows, the opening of those holes here, the burrows. That carbon dioxide is going to sink at the bottom and fill up those burrows. So you're actually going to cover it up with soil, and it kills those rats inside as well as the ectoparasites that are in them. So here's just a good example of some American cockroaches that were slipping out here once we started using the dry ice, but those, if you leave them in, they definitely will die. Trying to wrap up here for you today. It's been a long day, but look, here's some basic information again. I'm going to go back to this, and I want everybody really to think about their own area and say, hey, what information do I have here? And this is a process. You can't do it overnight, but you can do it over the course of years. And so again, set your priorities. for 1 year, 5 year, 10 year? Where is it you want to go so that your jurisdiction will be much better prepared if there is a rodent-borne outbreak? So between Bruno and myself, I hope that everyone here has seen that these commensal rodents aren't just a nuisance. A lot of folks think that, but they are not. They pose a very serious threat to human health and cause economic loss, right? They're excellent agents for disease transmission due to their adaptability and their opportunistic nature with humans. The thing with rodent control is there aren't very many reported human rodent-borne cases in the United States, all right? There are many cases in different parts of the world. And so again, if you are outside, you know you're in a rodent-borne area, don't wear sandals when it's wet, and that's a leptospira situation. You know, really be cognizant of what you're doing and what clothes you're wearing. Historical and current data supports the presence of a variety of diseases associated with rodents and their ectoparasites. All right, you can do the literature search. Vancouver, New Orleans, New York, it keeps on going. So very important. And so I always say this, and we need to be loud, is the local and state agencies must have the infrastructure to manage rodents in order to mitigate damage and disease. We must do better. So anyway, so I'm going to kick it over back to Astacia. I believe, hopefully we've got some questions in the chat. I haven't looked at it. So let's see, Astacia, do you have any questions?
Okay, yes, we do. We have some questions.
All right.
Does only rat dander cause allergies?
So mice, I know mice for sure, but it's different proteins that are shed off of the animals, okay? So I think dander is more like skin cells, but they actually do shed proteins. So yeah, I think that, no, so not just dander.
How can I get a copy or a link to the dumpster management flyer shown?
Absolutely. So we would love to give you that. So I believe we have already posted that at NOLA Mosquito on our website, on our social media feed, but you can also email us at education@nola.gov, and we'll make sure that we get it to you. With those different magnets and even the flyer, we had to really go in and research our state sanitary code, which, by the way, is excellent in the different, rodent-related topics that are enforced, as well as we have ordinances that are on the books for rodent control management and different issues. So those are two locations. You'll have to research your own to make sure that it's, everything complies with your area. Yep.
Have there been any studies on why humans seem to have an immediate adverse reaction to rats, but often think squirrels are cute? This reaction seems to be universal across cultures and innate, as opposed to learned behavior.
I know. So Mickey Mouse, right? So I've actually talked to, I don't know if there is a particular study on that. That's probably more sociology, so I don't know. But this is something that we actually, try to fight, this image of rodents, that they are so cute. Okay, well, look, a nice, clean rodent, house mouse or a nori rat, even a roof rat, yeah, I would say they're almost cute, right? But you have to really think about everything else that's associated. But Mickey Mouse, okay, Mighty Mouse, if you guys remember that cartoon. Chuck E. Cheese, right? That's another mouse. There are so many different mice that are associated with cuteness. But I think, again, as an industry, we need to really get the message out that they are a pest of concern.
What was the frequency in which rodenticide baits were checked and replaced?
Yeah, so great question, right? So I'm assuming that's going to be after Hurricane Katrina. So we were out there every 2 weeks. We were getting quite a bit of feeding. You saw that, my slide, didn't spend a lot of time on it, but every 2 weeks or so we were going. And when it was after COVID, right, we started at every 2 weeks. And then as the activity started to back off, we were able to get it down to a monthly check. And then when we didn't need it anymore, we actually replaced it with non-toxicant bait in some locations. And actually, we're revisiting all of that now. We just had an internal meeting this week. And we're actually going to be removing some bait in stations in some of our locations.
Okay. Can you please talk more about using bleach in trash bags? Does it just mask the smell of food?
Right. So that's what we were trying to do, right? So the whole goal here is that if you leave, we'll say this, let's just set it up. If we leave trash bags on the street, or at least trash bags in vulnerable placements, it may be an alley, it may be in a trash room, in a facility, you're really providing the opportunity for these rodents to smell those food odors, right? And eventually get into those trash bags. And so, yeah, I mean, part of it is to, I wouldn't say mask, but that chlorine, and it's a 10% solution that we were using. And actually we ended up spraying a lot of bleach on the bags, but we were, still 10%, but with a homeowner, a couple of squirts within a bag, 10% is good. And so that chlorine smell is quite pungent. So I don't know if it would necessarily mask it, but rodents don't like it. I don't know if that helps answer that question.
That seems to be the last question, just double checking.
Great.
Yeah,
okay.
I don't see any more. Oh, wait, there's one more.
All right, one more.
Why are you removing some of the rodent stations from certain parts of the city?
Yes, look, so some of these areas that we've been working in, so when COVID happened, it actually was 2 years yesterday that the first case in Louisiana occurred. So, so we have been monitoring many of those for a long time. And, if the areas were not active anymore, or we had very little activity with rodents, there's really no reason to leave those stations and that bait there. So anytime you're working in a particular area, you have to continue to inspect in service. The other part of it too is that we have lots of spaces that people are back. And so if you leave these stations on a sidewalk or in a very not a concealed space, they get kicked around, they disappear, all kinds of things like that as well. So obviously, if the area is very active, we're going to find more hidden locations to move those stations. But I mean, if an area has been inactive for a long time, there's no reason to have like a row or a street with bait stations. Now, what we are going to be doing is reassessing those areas again, right? Saying, okay, let's find locations, our sentinel locations, that we're going, maybe hidden away in a back alley somewhere, that are not going to be very disturbed, where we can go and check those on a regular basis as part of our surveillance activities. So that's what we're doing now, is really trying to understand how to set up a meaningful surveillance system. So somebody asked about pet rats, right? So I know you don't get it, but look, rats are really interesting. They're complex. A lot of people keep Norway rats and other rats as pets. I've heard they actually make good pets. We actually have a couple in our building that we use for our pets, that we use for demonstration purposes and education. So again, you have to leave everything clean. Make sure those animals are well taken care of, fed properly, exercised. They're quite complex. All right.
Okay. I think that's the end of the questions. Just want to invite everyone to follow us on our social media platforms, Facebook, Twitter, and Instagram. We're at NOLA Mosquito. Our next 4-part education seminar series will be on termite biology and control, and that starts Thursday, April the 7th. That's going to be a lunchtime series, so it'll be from 12 to 1:30 p.m. And once again, it'll be 4 parts just like this one. Just want to thank everyone again for attending today's webinar. We will be posting this webinar on our YouTube channel. If you're local and you have any rodent issues, please call 311 to report them. Please email any comments or suggestions to education@nola.gov. And this presentation is to be used for educational purposes only. All products shown are for educational purposes and does not constitute an endorsement by New Orleans Mosquito Control.
All right. Thanks, everybody. Thanks for taking the time this afternoon, and have a great day.